Pharmacognosy of cinchona. Synonyms: Cortex cinchonae, Peruvian or Jesuit's bark. Biological source: cinchona is the dried bark obtained from the stem or the root of Cinchona calicea, Cinchona ledgeriana, Cinchona officinalis, and Cinchona saxirubra, or hybrids of any of the first two species with any of the last two species. Family: Rubiaceae. Geographical source: tropical valleys of the Andes, Bolivia, and southern Peru. Cinchona is a native of South America, occurring wild there. At present, it is mainly cultivated in Indonesia, Zaire, India, Guatemala, Bolivia, Ceylon, etc. History: the name Cinchona is said to be derived from a countess of Cinchon, wife of a viceroy of Peru, who it was long believed was cured in 1638 from a fever by the use of the bark. The further distribution of the bark was largely due to the Jesuit priests, and the drug became known as Jesuit's powder or Peruvian powder. It first appeared in the London Pharmacopoeia in 1677 under the name of Cortex peruanus. Due to the increased demand for the tree, its cultivation was tried in various parts of the world like Europe, Java, India, etc. The cultivation in Europe was totally unsuccessful, while the cultivation those species grown in India like Cinchona saxirubra and in Java Cinchona ledgeriana were very successful. Today, India exports cinchona for more than 1 crore. Cultivation and collection. Cinchona is propagated by seed sowing method. The seeds are sown in boxes and the seedlings are transplanted to nurseries when they reach a height of 5 cm. The seedlings grown in shade till they attain a height of about 25 cm. When the plants are about 1.5 years old, they are transplanted to open space at a distance of 1 meter into well-drained, rich and porous soil. Cinchona grows well at an altitude of 1500 to 2000 meter above sea level, temperature ranging from 10 degrees centigrade to 30 degrees centigrade, and an annual rainfall of 200 to 400 centimeter. The plant is allowed to grow till 6 years, and then the first crop is collected by copicizing, uprooting, or by felling method. The bark is collected till the plant is 9 years old because the alkaloid content in the bark decreases thereafter. They are then dried under sun or by artificial heating by providing gentle heat. During drying, the bark attains quill shape and the color changes to red or brownish red. So this is the morphological characters of cinchona. Color: the outer surface is yellowish to brown with short fractures, and the inner surface varies in all the 4 species. Like in Cinchona calicea and Cinchona ledgeriana, it is yellowish, and Cinchona officinalis is slightly brown, and Cinchona saxirubra is reddish brown. Odor: distinctive. Taste: it's highly bitter and astringent. Shape: curved quill or double quill. Size: 30 cm long and 2 to 7 mm thick. Extra features: the outer surface consists of longitudinal and transverse cracks, fissures, ridges. Microscopical characters: the transverse section of bark shows cark composed of uniform layers of thin-walled cells containing amorphous reddish brown matter. So this is carc. Below carc is the region of cortex, composed of tangentially elongated parenchymatous cells with red, brown and thin walls containing small starch grains. Idioblast, containing microcrystals of calcium oxalate and secretion cells are scattered in cortex. Phloem consists of compressed and collapsed sieve tubes, phloem parenchyma, similar to cortex, and irregularly arranged, large, spindle-shaped, lignified fibers are present. So these are the lignified fibers which are shown in different color. Okay, so they are phloem fibers. Medullary rays are narrow, 2 to 3 cell wide and almost straight. Longitudinal sections of bar shows brick-shaped cells of medullary rays, longitudinally elongated cells of phloem parenchyma and fibers with visible pits. So this is the ground plan. So the upper layer is the carc, phellogen, phelloderm, cortex, secretory cells, idioblast, medullary rays, and phloem fibers. And this is the isolated phloem fibers. The length of the fibers are bigger when compared with the cinnamon. That is one of the identification character of cinchona. Chemical constituents. More than 30 alkaloids have been reported in cinchona. The chiefly identified alkaloids are quinidin, quinine, cinchonin, and cinchonidin. These alkaloids are the stereoisomers of each other, like quinine is stereoisomer of quinidin and cinchonin is stereoisomer of cinchonidin. Quinine and quinidin has a methoxy group in it, but cinchonin and cinchonidin do not have a methoxy group. The other constituents available are quinarenin, cincotin, hydroquinone, hydrocinchonidin, cinchotanic acid, etc. Other than these, it also consists of bitter glycoside, starch grains, calcium oxalate crystals, and crystalline acid like quinic acid. This is the structure of quinine and this is quinidin. Both are stereoisomers. Okay, only the changes in their arrangement. Similarly, cinchonin and cinchonidin are stereoisomers. Only the spatial arrangement, that is the configuration arrangements alone, will differ from each other. Coming to the chemical test, Thalioquine test. To the extract of cinchona powder, add 1 drop of dilute sulfuric acid and 1 ml of water. Add bromine water dropwise till the solution acquires permanent yellow color and add 1 ml of dilute ammonia solution. This gives emerald green color. That shows the presence of cinchonine components. The powdered drug, when heated with glacial acetic acid in dry test tube, evolves red fumes which condense in the top portion of the tube. Cinchona bark, when moistened with sulfuric acid and observed under ultraviolet, shows a blue fluorescence due to the methoxy groups of quinine and quinidin. Uses. It is mainly employed as anti-malarial drug for the treatment of Plasmodium falciparum infections in the many areas where the organism is now resistant to chloroquine and other antimalarials. It is also used as analgesic, antipyretic, protoplasmic, bitter stomachic and tonic. Quinidin is employed for the prophylaxis of cardiac arrhythmias and for the treatment of atrial fibrillation. It also has antimalarial properties and, like quinine, is effective against chloroquine resistant organisms. Cinchonidin is used in rheumatism, and neuralgia. Substituents or adulterants. Cupra bark belongs to the family Rubiaceae. It differs in its morphological character with cinchona but consists of constituents like quinine, quinidin, cinchonin, cinchonamine, etc. The other species of Ramigia, that is Ramigia purdiana or false cuprabar, does not contain quinine. These are the substituents or adulterants of cinchona. Thank you.